Hi guys, my name is Soo Hyun Im from University of Constance. This is a flash talk for my poster, Identifying Aureochrome-promoter Interactions in Phaeodactylum tricornutum. Let's start with a brief introduction about diatoms. Diatoms are unicellular microalgae that is known to contribute about 40% of global primary productivity. They are widespread all over the world, and one of the main reasons why they could thrive in everywhere is that they have evolved sophisticated photoreceptors to acclimatize to surrounding environments. Among these photoreceptors, we've been paying attention to a blue light photoreceptor, Aureochrome, because of their unique features. Aureochromes are similar to phototropins, which are blue light photoreceptor in green lineage, in terms of their domain composition of the protein. Aureochromes possess love domain to sense the light and B-zip domain as an effector domain, which can form dimer, then directly bind to DNA and function as a transcription factor. We're using the diatom Phaeodactylum tricornutum to study aureochromes, and there are four isoforms: Aureochrome 1A, 1B, 1C, and 2. So far, among these four aureochromes, Aureochrome 1A has been studied most intensively. Aureochrome 1A has been postulated to function as a master switch because 90% of genes were not regulated in Aureochrome 1A knockout mutant upon red light to blue light shift condition. We came up with some assumptions how Aureochrome 1A can affect such a huge range of gene expression. First, there must be cooperative functions among aureochromes. We know that they can form homo or heterodimers, and we think different combinations of aureodimer can affect a lot of mechanisms in the cell. Second, Aureochrome 1A can affect other photoreceptors, as well as other transcription factors, which can regulate gene transcription that leads to the downstream signal cascade. To confirm these assumptions and really figure out which genes are directly regulated by aureochromes, we used yeast-1 hybrid system. This system is used for identifying protein and DNA interactions. We inserted two vectors in the yeast. One makes the aureochrome protein, and the other one was for promoter site of the gene as a DNA base sequence. When the yeast sample is ready, cells are spotted onto the selective agar plate in serial dilution. If there is an interaction between prey protein and DNA base sequence, they can grow on the selective agar plate. First, I checked the interactions between each aureochrome protein with different aureochrome promoter sites. C0 was used as a negative control, which doesn't have any aureochrome protein. And yeast construct having aureochrome 1A protein with cyclin 2 promoter site was used as a positive control since this interaction was already confirmed by Huisman et al. Aureochrome 1A and Aureochrome 1B protein showed a positive interaction with Aureochrome 1A, 1C, and cyclin 2 promoter sites, while Aureochrome 1C and Aureochrome 2 protein didn't show any interactions with any of promoter sites. Also, I checked if Aureochrome 1A protein can directly regulate the ritmo genes. Ritmo protein is one of the transcription factors that is known to regulate deal biological rhythms in Phaeodactylum tricornutum, and it's assumed to be affected by Aureochrome 1 protein. Interestingly, there was no interactions between Aureochrome 1A protein and ritmo promoter sites. Here's my summary and conclusion. We verified the self-regulation of Aureochrome 1A. Aureochrome 1A showed a direct interaction with the promoter site of Aureochrome 1A, 1C, and cyclin 2 gene, and same goes for Aureochrome 1B protein. Since all the interaction assay was done in dark condition, we concluded that Aureochrome 1A and Aureochrome B protein can function as an activator for gene transcription independent of the light condition. Genes that are directly regulated by Aureochrome 1A are assumed to have an impact on the downstream signal cascade for the overall gene regulations. Based on these results, we could gain a better insight about the function of aureochromes on gene regulations. Thank you so much for your attention, and if you have any questions or suggestions, please feel free to contact me. Thank you.